Okay. Hi, my name is Andrea Jones. I'm a senior at the University of Oklahoma. And this summer I spent time in the Nelson lab studying soybean domestication, specifically what genes are differentially expressed between the wild and cultivated species. So why do we study soybeans? They're very agriculturally and economically important as they're a rich source of protein. So it's good for a lot of human foods, livestock feed, etc. And then the difference between cultivated and wild is that wild soybeans have never been domesticated or touched by human hands, while the cultivated soybeans have been selectively bred for characteristics like node development, flowering time, pod size, etc. And specifically, you want to study root architecture. So cultivated soybeans have more lateral roots than wild soybeans at the same time point. As you can see in this figure here, it's a little hard to see. And then also wild soybeans have delayed root development compared to cultivated. So you can see there's a lot less lateral roots here at 5 days versus there at 5 days. And for the experiment, so this part of the experiment was done by our collaborators in Pakistan, while this part was done by me this summer. So basically, they collected the samples at these time points in tissues, and then they sequenced them using Illumina sequencing. And I got the raw data in June. So I checked the raw reads for the quality using FastQC. I cleaned all the adapters off of them. And then I aligned a clean reads to genome using a program called HiSat2, which gave us this BAM file that we can use in the next program called Feature Counts, which counts the amount of genes in each replicate. So we have 18 files of that, 18 files of that. We paired them together and compared them as the cultivated as a treatment and then the wild as a control. And in this program called DESeq2, it can do a PCA analysis, which checks the quality, as well as give you a spreadsheet of all the differentially expressed genes and their log two-fold change. So for our results, we have these two hierarchical clustering plots, which basically shows that within each condition, the replicates are very similar to each other, which means our samples are good quality, highly similar to each other. We don't want like 10 hours after germination to be very similar to 5 days. And then next we have this Venn diagram showing the differentially expressed genes in different conditions. So basically so each of the tissues, these are specific only to that tissue, that time point. However, the ones in the middle, the 1769 genes, they're conserved throughout the whole growth of the plant. And so those are the ones that we want to study. And of those 1769, 35 of those code for transcription factors. And then finally, we have a gene ontology enrichment analysis, which is basically, we use this program called ShinyGo. And basically it has like a database of all genes, and pretty much all the gene IDs have certain pathways associated with. And so this program takes a list of upregulated genes that I generated from the spreadsheet, and I put them in, and it basically shows that pathways related to fluid and water transport are highly enriched in this whole sample here at the 5 days lateral root sample. So obviously, if it has to do with fluid water transport, maybe it's that's why the roots are just growing so much more in the glycine max compared to glycine soja. So for our conclusions, domestication has had an impact on the genes controlling root development. The shared DEGs might be conserved genes here, causing the phenotypic differences in the root system. And based on geoannotation, genes associated with fluid and water transport could be the reason of extensive lateral root system. So for future directions, we want to investigate the 35D transcription factors, and then also maybe do a functional analysis of candidate genes by using CRISPR in soybeans or Arabidopsis.
That's such exciting research. Sounds like you've been really busy then working with a lot of data this summer. Wow. Okay. So I have a question just from your introduction to this. So I've heard some great talks already today about, domestication versus wild relatives. And so I'm curious if the wild soybean has any advantage compared to the domesticated, or if there's anything there that you think would be interested in kind of going back to.
Yeah. So I guess wild and cultivated, they have their own advantages in different areas. So for example, for human use, the cultivated would be better. But for if they wanted to exist with little water in the environment naturally, glycine soja would totally be better. It probably has more drought resistance due to like it having like a lower overall need for water. Glycine max, the cultivated, they breed it for like the large amount of pods, large amount of flowers, extensive roots. So it obviously takes up more water. So I would say the biggest advantage they have around each other is water usage.
Makes sense.
Yeah.
That's so interesting. And was there anything that came out of the Go enrichment that surprised you at all that you weren't quite expecting?
I don't think so. I mean, it'd be cool to see different. I had some at different time points. They were like a response to like mechanical stress or disease pressure, but mostly they were fluid and water transport, which I expected.
That makes sense. Thank you so much for sharing all the hard work you've been doing. It's amazing. And we wish you the best in the future. Thank you.